You know when you see one of those headlines, new study discovers X, and you look closer and they only studied like 50 people? And you're thinking, how in the world can they know that for sure? It seems kind of impossible, doesn't it? Well, there's a statistical secret behind it all. It's how scientists take tiny little samples and make these huge, confident claims. And that's exactly what we're going to break down today. I mean, this is really the question, isn't it? It honestly feels a little bit like a magic trick. How can 100 people in a study tell you something that's true for millions? It just doesn't seem to add up, but that's the puzzle we're going to solve. Let's figure it out together. All right, so like any good mystery, this one starts with a clue, and our clue comes from a place that, let's be honest, can be super dense and confusing, a results table from a real research paper. Let's go ahead and zoom in on this one little piece of the puzzle. Okay, so you're looking at this wall of numbers, right? And one little thing just jumps right off the page. Sig equals 0.000. That's it. That's the researcher basically sending up a signal flare, screaming, hey, I found something real over here. But what does that actually mean? How can 0.000 be a good thing? To crack that code, we first have to wrap our heads around the biggest challenge in pretty much all of research. And that brings us right to the heart of the matter, something called sampling error. This is the whole reason we can't just look at that 0.000 and call it a day. You can think of it as, well, it's kind of like the ghost in the machine of research. Okay, so first things first, let's get our terms straight. You've got the population. This is the entire group we care about. We're talking every single university student in the country, or all the fish in a lake. It's the whole shebang. We want to know the truth about them, but here's the problem. You can't. It's impossible to study every single one. So what's the workaround? Well, we take a sample. It's exactly what it sounds like. It's a small, manageable chunk of that bigger population. The photo analogy's perfect. It's like you're taking one picture of a giant crowd, and you're just hoping that your little snapshot gives you a good idea of what the whole crowd looks like. And this really gets at the fundamental gap we're trying to cross, right? On one side, you've got the population. That's the whole truth, the thing we really want to know. But on the other side, you've got the sample. That's the little piece of data we can actually get our hands on. And the big challenge? The sample can totally be misleading just because of random luck. Okay, so this gap, this natural, totally unavoidable difference between our sample and the whole population, that's what we call sampling error. And listen, this part is so important. It's not a mistake. Nobody messed up. It's just a product of pure random chance. Think about it. If you grab 100 people at random, is their average height going to be exactly the same as the average height of all 8 billion people on Earth? No way. It'll be close, maybe, but it's going to be off a little, just by the luck of the draw. So that brings us right back to our central dilemma. When we see something interesting in our sample, say one group scores higher than another, we have to ask the big question. Is that a real thing that's happening out there in the whole population? Or is it just a fluke? Is it a ghost in our data created by that random sampling error? We need a way to tell the difference. We need a toolkit. So how do we do it? How do we bust this ghost of random chance? Well, luckily for us, statisticians have built this incredible detective kit. And all the tools in this kit are designed to do one basic thing: measure exactly how weird, how unusual our finding really is. And the first tool in our kit is this thing right here, the normal distribution, or as everyone calls it, the bell curve. Think of it as a map of what's normal. Stuff piled up in the big hump in the middle, totally common, totally expected. But the stuff way, way out in those flat little tails, whoa, that's the rare stuff. That's the surprising stuff. Okay, so we have our map, but how do we find our exact location on it? For that, we use something called a z-score. Z-score is like a universal translator for your data. It doesn't matter what you measured, test scores, height, weight, the z-score converts it into a standard language. It tells you exactly how far from the average your result is. It's literally our measurement for weirdness. Let's make this super concrete. Imagine you take an exam, and the average score for everyone was a 65. You open your results. You got a 54. Ouch. But is that like really bad or just kind of bad? Well, let's find out. We take your score, subtract the average, and divide by the standard deviation. Boom. We get a z-score of negative 1.22. We can then look that up on a table, and it tells us the probability is about 11%. That means only 11% of people did as poorly as you or worse. See what we just did? We took a raw score and turned it into a precise number that tells us exactly how rare it is. All right, so we've got the tools in our kit. We can measure unusualness. Now let's look at the actual formal process, the official rulebook that researchers follow. It's called the hypothesis test. And honestly, the best way to think about this whole thing is like a courtroom trial. I love how Josh Starmer from StatQuest explains it. You start with the default assumption, basically innocent until proven guilty, and then you see if your evidence, your data, is strong enough to get a conviction. So in statistics, this presumption of innocence has a name, the null hypothesis. You can think of it as the ultimate skeptic. It's the default idea that there's nothing going on. No effect, no difference, no relationship. The null hypothesis says that anything you think you see in your data is just a random fluke, just that pesky sampling error again. And the entire goal of your experiment is to collect enough evidence to prove that skeptic wrong. Now, in our courtroom, we need a standard for beyond a reasonable doubt. And we have to set this before we even start the trial. In stats, this is called the significance level, or alpha. The standard is usually 0.05, or 5%. What that means is we're willing to take a 5% risk of making a mistake, a 5% risk of convicting an innocent person, of saying there's an effect when there actually isn't one. It's our line in the sand. And now for the star witness, the key piece of evidence in our trial, the p-value. Okay, now this is the most important definition, so lean in. The p-value asks a very specific question. It asks if, and that's a huge if the null hypothesis is true and there's really nothing going on, what are the chances we'd see results as extreme as the ones we got just by pure random luck? So think about it. A really, really small p-value means your result is a super unlikely coincidence. It's a shocking piece of evidence against that presumption of innocence. Okay, the evidence has been presented. Our star witness, the p-value, has spoken. It's time for the jury to make a call. And all we have to do is take our evidence, the p-value, and compare it to our standard of proof, that line in the sand, alpha. And the decision rule could not be simpler. It's this. If your p-value is smaller than your alpha, if the evidence is stronger than your standard of proof, you reject the null hypothesis. The verdict is in, statistically significant. But if your p-value is bigger than alpha, the evidence just isn't strong enough. You can't reject the null. The verdict, not statistically significant. That's it. All right, let's go all the way back to the beginning. Remember our first clue? That mysterious little sig equals 0.000 from that table? Now that we have our detective kit and our rulebook, we can finally crack this case. So it turns out, sig is just another name for, you guessed it, the p-value. And a p-value of 0.000, that means the probability of getting this result by pure dumb luck, if there was actually nothing going on, is less than 1 in 1,000. I mean, that's not just unlikely, it's incredibly unlikely. It's way, way smaller than our 5% line in the sand. So, what's the verdict? We reject the null hypothesis. We have to. This isn't a fluke. The researcher found something real. So there you have it. We've cracked the code. We now know how researchers can look at a small group of people and use the power of probability to tell if what they're seeing is a real discovery or just a random fluke. But that leaves us with one last, really important question. Just because something is statistically significant, does that mean it's actually important in the real world? You could find a new drug has a statistically significant effect, but maybe it only makes people better by 0.1%. Is that really a breakthrough? Learning to tell the difference between what's real and what actually matters, well, that's the whole next step in thinking like a scientist.